I'm Sarah, and thank you very much, Dr. Chambers and the Data2 Foundation for inviting me to speak today. The research that I'm going to share with you today begins for us in 2018 with the identification in siblings of a biallelic variant, C1052T to A in exon 7 of the ADA2 gene. And this variant results in a leucine to glutamine substitution at amino acid 351 in the catalytic domain of ADA2. So using serum of the older sibling, which is labeled on my slide as P6 in the graph on the left, we demonstrated that leucine 351 to glutamine was associated with low but not abolished ADA2 enzyme activity. And this is consistent with other cases of childhood onset DATA2, and in contrast to ADA2 activity in sera from healthy children, as well as in children with vasculitis that did not carry rare ADA2 variants. And this assessment of ADA2 enzyme activity in patient sera or plasma is performed in our laboratory with a modified version of the colorimetric diazyme adenosine deaminase activity assay. So I think there's more of us out there that use this one, but I'm showing it in the middle panel. And we've actually published normative ranges of ADA2 enzyme activity in children using this assay, and we routinely rely on it in our lab for research, as well as in conjunction with ADA2 gene sequencing. We're using it to support DATA2 testing in our province. So notably, the siblings with the L351Q variant, they had quite early and severe disease presentation. So symptom onset in the younger sibling was at 1 week of age with suspected Diamond-Blackfan anemia and subsequent development of severe neutropenia. So that's what's shown in the panel on the right. There was no evidence of neutrophils or precursors on a bone marrow biopsy. And unfortunately, this child succumbed to their disease at 17 months of age before they could receive appropriate treatment. Their older sibling was asymptomatic until age 3 years, when they presented with a similar picture of neutropenia, fevers, and evidence of prior stroke. And with the knowledge of their sister's diagnosis, this child was treated with a BMT. We know that neutropenia has been reported in approximately 10% of individuals with DATA2, and was also observed in the CECR1B knockdown zebrafish. But two particular reports of DATA2 caught our interest due to similar presentation of neutropenia and identification of variants. So the tyrosine 353 histidine variant and the glycine 321 glutamine variant, they're in AD2, and in terms of protein structure, they appear to be in quite close proximity to leucine 351. So together, this has led me to my doctoral thesis research, which is investigating the roles of ADA2 in hematopoiesis, specifically with the focus on granulopoiesis, and then looking at the impact of region-specific variants on ADA2 structure and function that in turn might contribute to the severe hematologic defects that we're seeing or that are observed in some individuals with DATA2. I think we've seen the slide already, but towards this, we have established an in vitro system to culture and differentiate CD34 positive hematopoietic stem cells, both sourced commercially, and then I'm really lucky we can source them through our local cord blood bank towards monocytes and granulocytes with an initial aim to characterize temporally ADA2 expression during development of these cell lineages. So I'm using multicolor flow cytometric analysis of cell surface markers, and I can track over a 14-day period of differentiation a shift in the population from CD34 positive HSCs to CD34 negative CD14 positive monocytes, which are shown in the upper left-hand quadrant of the scatter plot on day 14 of differentiation. And similarly, we have the shift of CD34 positive HSCs to CD34 negative, CD15 positive, CD11B positive granulocytes. So that's shown in the scatter plot on the right. In parallel, what we're able to do is track ADA2 catalytic activity. And we're using that as a surrogate for released ADA2 in the cell supernatant. Doing so, we see that enzymatically active ADA2 is secreted by HSC-derived monocytes detected by day 7 in our system. So you can see the dot coming up by day 7. And it continues to accumulate as the cells differentiate further. Perhaps not surprising based on what we know about mRNA expression of ADA2, HSC-derived granulocytes, which are cultured in an equivalent concentration to the HSC-derived monocytes, express negligible to small amounts of catalytically active ADA2 in our system. So moving forward, I have established methods in these CD34 positive hematopoietic stem cells to deliver Cas9 sgRNA ribonucleoprotein complexes to knock out ADA2 and introduce ADA2 variants, such as L351Q, in order to test the impact on lineage development, both in isolation and then in co-culture, so the monocytes and the granulocytes together. And in this system, we can evaluate the dependency of granulopoiesis on ADA2 expressed by the myeloid lineage, and explore whether hematopoietic deficiencies in DATA2 might derive from a similar or a different mechanism than what we've talked about and has been described for ADA1 SCID, which is the impairment of lymphocyte development attributed to the intrinsic accumulation of adenosine and adenosine byproducts by ADA1 deficient lymphocytes. So complementary to our work in the hematopoietic stem cells, we've established an in vitro expression system that allows us to more readily assess the impact of pathogenic variants such as L351Q, ADA2, on ADA2 expression, processing, and cellular function. And in this system, we engineered flippin CHO cell lines, which are naturally devoid of ADA2, to stably express 6 times his-tagged wild-type ADA2, L351Q ADA2, and as a comparator, ADA2 with a G47R variant that is common in DATA2 patients and frequently associated with vasculitis, not neutropenia. We evaluated secreted and retained ADA2 protein in the cell supernatant and lysate, respectively, by Western blotting for the 6 times his-tagged proteins, just shown on the left, and quantitated by densitometry analysis in the graph on the right. And as expected, wild-type ADA2 and ADA2 carrying the G47R variant was detected in the extracellular space. In contrast, we discovered that the L351Q ADA2 variant, as shown by the red lines and bars, was expressed but was not secreted in any appreciable amount. So the bands corresponding to ADA2 were detected in the lysate, but not the supernatant. And we also did densitometry analysis, which revealed a similar quantity of expressed wild-type and L351Q ADA2 protein, but an 8-fold greater intracellular retention of L351Q compared to wild-type ADA2. And in addition to what are approximately 60 kilodalton monomers, noteworthy would be the presence of L351Q ADA2 dimers at approximately 120 kilodaltons that were not observed in comparable abundance for retained or secreted wild-type ADA2 or G47R ADA2 protein. Naturally, we were wondering, maybe you weren't wondering, but we confirmed that the G47R variant and the L351Q variant were damaging to ADA2 enzyme activity, as you would have expected. And then we went on to question whether this intracellular accumulation of L351Q ADA2 was having any sort of negative impact on the CHO cells that could potentially promote a state of cellular stress or might contribute to an inflammatory phenotype. And we found no evidence based on LDH, or on the release of LDH, reduction of MTT, and CFSE cell tracing to suggest that intracellular accumulation of L351Q ADA2 confers a change in cell metabolism, toxicity, and/or membrane leakage. Similarly, there were no indications of ER stress in our CHO cells expressing L351Q versus wild-type ADA2. So that's based on the similar abundance of GPR78, which I'm showing there at approximately 75 kilodaltons in the immunoblot on the left. And I think, well, GPR78, it's a constitutively expressed, predominantly ER localized protein, and it facilitates elimination of proteins as part of the unfolded protein response. We did try to look at kind of the localization of ADA2 by confocal microscopy, and we were not successful, and we didn't have the methods or the techniques on hand to be able to do that well, so it would be interesting to see where this L351Q82 was in particular within the cells. Granted that we're working with CHO cells, which are not necessarily the model you want to be delving in too deep with. Ideally, we switch to a human cell line. Instead, what we did observe with the semi-quantitative PCR was elevated expression of type 1 interferon response genes, so that's IFIT3 and IRF7. They were up in resting CHO cells, which are shown in the bar graphs on the left, and type 1 interferon-stimulated CHO cells, which are shown in the bar graphs on the right. And so those cells that were expressing L351Q82 compared to wild-type 82 had higher expression of these interferon response genes. And although we didn't have the opportunity to measure the expression of type 1 interferon-stimulated genes in the siblings with the L351Q82 variant, these results are consistent with the reports of elevated type 1 interferon signaling in DATA2 patients and a spontaneously upregulated type 1 interferon beta response in 82 siRNA knockdown of U937 cells and HUVIC cell lines, as described by Dr. Sharma's group, or as described by Dr. Sharma earlier. The intracellular retention and accumulation of L351Q82 in our flippin CHO expression system is consistent with prior reports by Navin Elkan et al. Of variant-specific differences in 82 protein secretion and retention by transfected HEC293 cells. In that report, which is in the middle there, R169Q82 was near absent extracellularly, and a corresponding increase in R169Q82 was present intracellularly. Moreover, in our flippin CHO expression system, the amount of extracellular G47 R82 was approximately half that of wild-type 82, which is consistent with the observation by Zhou et al. Of a substantially reduced extracellular and intracellular 82 protein in macrophages from DATA2 patients followed at the NIH, which is shown there. As a closing thought, and just prior to my conclusion slide, this point may have already been made in part for me by our earlier discussion, but I think it's really important to recognize that gaps still remain in our understanding of the processes that govern 82 retention, secretion, and dimerization, as well as the impact that specific variants have on these processes. And related to that, it's our experience that there is a lack of tools to detect 82 protein. So we have had varying success with Western blotting for 82, with what are the commercially available primary antibodies in cell supernatants and lysates, and have tried, but unsuccessfully, to detect full-length 82 in sera and plasma with a commercial 82 ELISA. It's evident from the published literature that many of us rely on the introduction of detectable tags such as HIS or FLAG or MYC to monitor 82 protein, or use measures of catalytic activity as a surrogate for protein abundance, which has obvious limitations, not the least of which would be that it's an inability to assess catalytically dead variants, or the presence of catalytically dead variants in 82. Thus, I welcome, either at the end of this session or anytime during the conference, a conversation about the development of improved tools to detect and study ADH2. In summary, the methods we have established for the differentiation of stem cells in vitro have allowed for a preliminary temporal characterization of 82 enzyme activity during cell development, and our results suggest that monocyte-derived 82 may be required for granulopoiesis. Using our CHO cell expression system, we find that the pathogenic variant L351Q in 82 impairs secretion and elevates type 1 interferon responsive gene expression. And I will leave you with the suggestion that these results support the conclusion that the impact of pathogenic variants in 82 may go beyond the loss of catalytic deaminase activity. And with that, I would like to say thank you very much to all the people that made this work possible, especially my supervisor, and thank you to you for listening.